is some eye-like structure that is called ocelli. Ocelli is also present in the juvenile of Sarnagasua. This is, however, in adult, that ocelli will disappear. This is some of the characteristic features of the Sarna species that is available in Northeast India, especially in Assam. And actually, identification is a very good art. And just I want to share with you that 7 new species has been described or discovered from our center. This is the credit goes to my student. One species that is called Bedis. The characteristic features of that species is they can able to take the color of their surrounding. If you put light screen, it looks like light. If you put some blue, it will show like a blue. They can camouflage their body color. Economically, they does not have any food value, but commercially it is very viable, and that is in Lower Assam it is known as somewhere it is known as Randonimas. It is very small in size. We have discovered or described one species from Goalpara, so we named it as Bedis ponceratnensis. This is new to science. Another species that is called Sistura means loach, from the cave of Shillong. That is around 1 kilometer from the entrance of the cave. That has collected from one of my students, that his name is Dr. Klur Mukim from Ledikin College. And the species is totally blind. It does not have any eye, because you know that inside cave there is no sunlight. If there is no sunlight, no need of eye. But bell is very powerful, which act as searching of the feet. We synonymized or we named it as a Sistura larketensis. Larket means a village named Larket, which is found, situated very near to that particular cave. They are taking care of this cave. So to give a honor to them, we synonymize it as a Larketensis. In addition to that, another species we have described from Kameng River from Arunachal Pradesh, that is called Sistura ribo. All are new to the science. In case of Arunachali language, ribo means loach. So we synonymize it as a loach. You can, in that particular fish, I just want to tell you that sexual dimorphism, that male and female, in case of Cyprinidae, we can, in case of Rohu Katla, that is easy to recognize male and female. However, in case of small species like Sistura, it is not so easy to recognize both male and female. But in that particular species, which we have collected from Kameng River, they have special characteristics in their mouth region, so that we can easily recognize both male and female. Another species that is we have collected from Khasi Hills, that is from Shillong, that is also Sistura. And in that particular Sistura, I just want to show you, the first time we showing the strip, long strip throughout the body. This is the characteristic features of that particular species. We have published it in Zootaxa. Another, this is the first time we are integrating both conventional taxonomy with the molecular taxonomy. Another species we have collected from Dima Asao, we just published only in this year, in the month of March. Silorhynchus. Yes, please. Anything, any queries, please? Silorhynchus nalongthai. Nalongthai is a Dimasa language. It's meaning loach. All are loach. We have collected from Deung River from Dimasa. Just I want to tell you to the student that all these fishes are not coming by saluting us. Our student has go to very remote places of Arunachal Pradesh, of Assam, Dimasa, Karbi Anglong, etc. to collect these species. It will never come to you. You have to go to, you need to go to the site of that particular species. Consecutively this year, we have published, though it is pandemic year, we could able to publish 3 new species from our department. Another Silurincus we have published from Kameng River that has published in Vertebrate Zoology. Vertebrate Zoology, this is also new to science. Total 7 species we have described so far from our center. And these are, is it visible? My slide is going on.
Yes, sir.
Yeah. Some, at least 7 to 10 species are in prey. They are still waiting discovery. Hopefully, we can able to describe it. This is for your information. And how we can able to, this is for the student only, how we can able to identify that species, because it is a new to the science. It is our contribution to the science, how it is possible. Already I told you that identification is a very good art. You can take it as a hobby. Though you are still you are in, when you are going to market for buying fish, always try to do that. I just want to tell you how we are describing species, what is the genesis to describe the new species. The first one is morphometric and meristic count. I'll show you. Then osteological evidence, the skeletal system. Then molecular divergence. Just I want to tell you. This is the diagram of a Cyprinidae species. Just for your information, when you measure from the tip of the mouth to the tip of the caudal fin, end of the caudal fin, this is called total length. When the tip of the mouth to the origin of the caudal fin, this is called standard length. In that way, we have to measure the species, each length, the eye diameter, opercular length, barbel length, everything. That is called morphometry. When you are counting the scale, spine in the fin ray, that is called meristic. How many unbranched rays are there? How many branched rays are there? Whether the fin are adipose or it has rifts, etc. On the basis of that, we can able to identify. We can able to make. We need to make a Excel sheet. At first, we have to count everything of that particular species. Then we go for another method that is called osteology. This I want to tell you, those who are from zoology, one method you can also try in your lab, that is called Eligirin preparation. Eligirin preparation, that is very easy to do that. If you need, I can able to give you the protocol. Take a full size, purchase one species from market and put in Eligirin. After a few days, after a few days, that all the skeletal system you can able to take it out from the flesh. All the fleshes will go and the skeletal system will appear. And through that skeletal system, you can able to see the differences between the species. What is the difference between the skeletal system? This is also a very good practical you can able to try in your laboratory also. This is very easy. It no need so much of sophisticated instrument. Only some glass jar is required and some chemicals is required. You can do in your lab also. The third one is molecular evidence. We can able to use some of the gene that is called mitochondrial gene, cytochrome oxidase I, cytochrome B, etc. To identify, you have to go to the molecular divergence or molecular characterization, the final stage, not in the initial stage. Initial, you have to go for morphometric and meristic count, then osteology, then molecular evidence. From that molecular evidence, you can able to tell everything of that particular species, which is also very much helpful to do the barcoding of a species. Okay, this is I am audible. Is it interesting or not? You are feeling bored?
No, sir. Interested.
No, sir.
Yeah,
Yes.
you can. Just I want to tell you that this is the mouth part of a cystura. It has a small slit in the lower lips. This is the distinguishing feature, and you can see the whole lips are cushion type because they have to adhere themselves in the hill stream because they are found in highly torrential river. So they need to attach themselves by these lips to get rid of that high current. You can see this cystura, one small slits are there in the lower lip. In addition, this is the characteristic features of a cystura species. In addition to that, when you are dissecting the air bladder, air sac, in case of cystura, it is bilobed. If it is trilobed, it is another species. This is called physocystura. You can see the difference. Here in the lips, small slits or incision is there. However, in case of here, physocystura, you can see a big gap is there in between the lips, the lower lips. In addition to that, you see the elongation of this lips. This is the some of the it is not easy to tell all the characters. This is some of the characters you can able to identify, and it is very easy to identify using magnifying glass and simple microscope. Another species that is called Aborigines. In case of cystura as well as physocystura, they are having 3 pairs of barbels. Here 2 pairs, but you can see the lips characters. You see the elongation of the lips character. This is a drawing. This is not the real picture. In that way, you can able to differentiate different species. Now, this is very interesting that Gara, the peculiar, I want to tell you, more than 300 cystura species are available in world, out of which 50% in available in Northeast India. This is for information. More than 30 to 40% cystura still waiting discovery. Still waiting discovery. In addition to that, Gara, it is also very peculiar. If you see the mouth part of a Gara, this is called proboscis. Gara is found 2 types, without proboscis, with proboscis. And the distinguishing feature is the mouth part. That is called oromandibular structure. You can see some cushion-like structure through which you can, this is the adhesive apparatus by which you can, this species can able to stay in a torrential river. By this oromandibular structure, the difference in the oromandibular structure, Gara species can able to identify. In addition to that, you can see the glyptothorax with these are having that is that ventral adhesive, that adhesive apparatus is the most distinguished features of this glyptothorax. This is called stri, that ribs which is coming. This is the distinguishing features of that particular species. It is also known as cat species. If you have any queries, anything to ask, you can ask, no problem. Hello, all the listeners, please, if you have anything to ask, you can ask. in between.
The participant friends, you can put your questions in the chat box.
Okay. Shall I proceed again? Okay. You can
Yes,
see
sir.
the adhesive apparatus. You can see in case of Glyptothorax, the structure. However, in case pseudoensis, you can see the structure. Another species that is called pseudolagobia, you see the adhesive structure. Just I want to tell you something morphological differences in between the peculiarity that differentiates species which is found in our region. And you know that all the barbel bearing species are known as catfish. Actually, catfish are not only having barbel. In addition to that, some other characteristics are also there. In case of catfish, in the pectoral fin, the first unbranched ray is very stiff. In case of magur, in case of singi, etc. This is also a characteristic features of catfish, with having 4 pairs of barbel, small eyes, non-proctectile mouth, etc. And in addition, in case of catfish, adipose fin are also there. Adipose means fin with flesh, no ribs. These are the some of the species which has been found. It is though it is looks like ugly, but it is enjoying very good price in case of international market. That is called Cretocyloglenis kamengensis from Kameng, Exostoma from also from Arunachal Pradesh, Myriglenis from also Arunachal Pradesh, etc. Okay, these are already I described that Silurincus, that Kamengensis has already been described. Just I want to tell you another, that is what is going on in our Assam or in Northeast India. That you know that Northeast India is regarded as the hotspot for ornamental fishes. Most of the freshwater ornamental fishes are going from this particular state only. Some illegal like rhino poaching, that fish poaching is also going on in our state. What is that? Most of the colorful species of our land is going to abroad by some way with through illegal trade, and these countries are getting the Singapore is getting 43% of freshwater species from Assam. Likewise, Thailand, United Kingdom, United Arab Emirates, Netherlands, Malaysia, etc. Germany, even in Germany also, they are getting 5%. That is from where we got this data. Recently, we concluded one project in collaboration with University of Oxford Department of Zoology. We just concluded from that particular project. Just I am sharing some of the data. What is happening in our without knowledge, without knowing our people, without the knowledge of our government. The target species already I told you that Sarna barca, Sarna orentimaculata, Sarna stewarti, Sarna blairi. This is the target species. In 2014, I just tell you from the data which we have gathered from that particular project. In 2014, if you remember, in the month of September and October, there was a huge flood in Assam. During that time, more than 10,000 Sanna barca has went to Singapore via Calcutta. During that time, the Sanna barca price goes down to 2,000 rupees. This is that data we have collected from that airport only. In addition to that, these are the peculiar species, the beauty of the fish diversity of Northeast India. Deru. Only 2 centimeter, that species is only, the adult size is less than 2 centimeter, but it enjoying 10 pound per pair. 10 pound per pair. Eructis cranisoi, that is found in Kokrajhar. Microfish Dakota, this is also found in Manas River Basin. These are the some peculiar fishes that is going abroad through illegal trade. And these are the country they are getting ornamental freshwater ornamental fish from our region. Okay. And through 5 designated airport, Kolkata, Mumbai, Chennai, Bangalore, and Kochi. These are the designated airport to which they can able to send the fish to abroad. How they are managing, I don't know, but they are every day they are sending. Just I want to tell you, showing you some photograph. You can see this is somewhere in Kokrajhar, in a remote place in Kokrajhar. You can see some people from West Bengal. They have also erected some of the cistern. Is it visible, Mitali?
Yes,
No,
visible,
no,
visible.
sir.
Yes.
Yes, sir, it's visible.
That
visible,
Your
sir.
page is not coming, sir.
Visible to me. It is visible
Pardon?
to me.
Yes.
It is visible to me. Presentation to pin kore diya, jar jar aha nai. Presentation to pin kore diya. It is in the screen.
Okay.
jar jar aha nai
Security
মই দেখা পাই
টাইম
আছো।
বেশি লৈ আছে
নাই নাই ঠিক আছে।
Security টাইম বেশি লৈ আছে
It is very interesting.
Yes, sir, your presentation is absolutely visible.
Okay. Okay, just I want to tell you that story that is still going on. That picture we have collected from Kokrajhar. You can see some of the cistern they have erected by some of the West Bengal people. The villagers, they are collecting the species from the wild, keeping in that cistern whole week. And every week, the vendors are coming and they are taking these live fishes by packeting through oxygen, using oxygen, and then taking it away by bus or by small vehicle. They are taking to Bagdogra or Siliguri. From Siliguri, they take it by train. That is going on. And I can show you another that photograph we have. That is for Sanabarka. This is a huge illegal trade that is going on in Assam. That we have collected that photograph very near to Guwahati University through spy camera. Our student has collected that photograph. So intentionally we make the person mouth blurred. And in that way, they are packeting the species with oxygen and send it to abroad. You can see the packet. That is, that villagers, they are getting 1 or 2 rupees, but these people are collecting lakhs of rupees from the ornamental species. Friend, you are the real people to stop this type of illegal trade. Otherwise, our diversity will finish in a particular day. Okay, just that I just if I am not boring you, then I want to show another topic that just we have concluded. If you find it is interesting.
Yes, sir, sure, sir.
Yeah. Okay. What is the major challenges to the ornamental fishes of that is fish diversity of Northeast India, mainly habitat degradation, over exploitation, alien species invasion, and water pollution? We don't have any river flow modification that is not done in case of Northeast India. 4 are the major challenges in case of Northeast India, out of which I will take one that you know that another hue and cry is going on in Upper Assam. You know that coal mining that is in Margherita and Lidu, some that government has already given permission to some traders to excavate coal from that particular sanctuary or not. I'll show you something like that. Okay, that you can see due to the siltation, some of the torrential river has converted into muddy from rocky, and this is a deleterious effect to the habitat or niche of the species. This is one of the fact. In addition to that, most of the tribal people in our Assam, including Meghalaya, in that tribal village, they don't have any traditional fishermen. They are always fishing through society fishing, and in that particular fishing, they are using herbal poison. Herbal poison. I can show you. This is in Assamese. It is called moinakata. It is also helpful in rheumatic pain. They are using the fruits of the. It looks like a citrus fruits. They are using the fruits, and by grinding that fruits, they just spread it and in the river to kill the fish. In that particular roots is so poisonous, even insects are also killed, or insect is also die due to the toxicity of that particular roots. I'll show you something. You can see these are the mahasir which has been collected from that one river from Garo Hills. Immediately after taking the fishes, they remove the viscera, that alimentary canal. Why they are remove the viscera? For two things. The first one, when they remove the viscera, the poison will not spread to the body, number one. The second one, when you remove the viscera, the fishes will remain intact for long time because they don't have place to store it. You can see that loaches, that Sistura, what I am saying, they are drastic, that 1 ton killing of this Sistura by using herbal poison. More than 30 herbal poisons are there in Northeast India, more than 30. Dynamiting, it is done in Dima Asau, crude bomb. They are using the powder of crackers and using some bamboo to ignite it. Put this dynamite, some cracks of the stone, and just blow it. The blowing is so powerful, even the big boulder also smashed due to that particular dynamiting. That is also still going on. In addition to that, this is very destructive fishing method. I am showing that particular person. If you see that particular photograph, that person is.
So sorry to interfere. Slide Hanai.
Yep.
Hanai. Nice, sir.
No,
Okay.
slide is showing. Slide is showing. It's okay, sir.
Hello.
Slide is showing.
Hello. যাৰ slide আহা নাই সেইটো তেওঁৰ নিজৰ প্ৰবলেম হৈ আছে থাকে। তাৰ মানে গোটেইখিনি পাৰ্টিসিপেন্টৰ য'ত সৰু সৰু স্লাইড আছে তাতে গৈ পেলাই মেইন প্ৰেজেন্টেশনটো তাতে ওলাব, তাতে গৈ পিন কৰি ল'ব লাগিব। আমাৰ তাতে স্লাইড আহি আছে।
আপনি দেখা পাইছেন না সকল?
Sir, please carry on.
স্যার আপনি গৈ থাকক।
You can see that person is carrying with bicycle. He is carrying one small battery. In the handle, he has one inverter. That capacity of that inverter is around 800 EB.
Sir,
800 watt.
Showing the presentation is not visible.
Please keep silent. Presentation is visible to all others, so please keep silent. There may be some network problem in your area.
Okay, to where that wire is coming from that particular inverter to, and this particular person is holding that wire. When it dip into the water, immediately that particular person, he just rang the bell, and whole area is that electrical current pass into that particular water. As a result of which all the fries, fingerling, etc. will electrocuted and they are coming out from their hide. This type of destructive fishing method is going on in our area. In addition to that, just I want to tell you, because sorry, one that is hue and cry is going on due to that coal mining in the Margarita and Lido area. What is happening in what is there in coal mining? What will be the effect of coal mining? I just want to tell you. Actually, I have been associated when I was in Gwalpara College. I have been associated with one river that is in East Garo Hills, that is called Simsang River. You can see the aerial view of this Simsang River. This is the longest river in Garo Hills. And here, somewhere here, that the famous national park that is called Balphagram National Park is located. That river flowing from 1400 meter MSL to 360 meter MSL. The river harbors, it says that villagers, though it is biologically it is not true, that they thought that electrical eel is present in that particular river. That electrical eel is known as Sodoria khajurial. It is looks like our bami mas, but it does not have any electrical organ. Okay, you can see in that both the bank of this river is harbor huge natural resources. You can see the jungles or that canopy because of this high diversity of flora. And in addition to that flora and fauna, to both the bank is a reservoir of coal. I can show you that coal mining in that particular area is done by a crude method that is called red hole mining. I have taken this photograph in 1992, but excavation takes place in 1986. You can see which is looks like that excavation takes place yesterday only. What they are doing in that particular area, that is in very near to Dudno, you can that Nagal Bibra. What they are doing, actually, in case of Garo Hills, then Garo people, the village leader is the owner of a particular hills, and some merchant from Dhubri and Gwalpara, they went to that particular place and they purchased that hills with a sum of rupees 10,000 to 20,000 to check whether any coal deposits are there or not. And that village, Gaubura, he sold that small hills at the rate of 20,000 or 25,000 or like that. That what the merchant did, they just removed the topsoil. This is just a destruction of the habitat. He just removed the topsoil to see whether any coal deposits are there or not. If coal deposits are there, then bargaining again start from that excavation of that coal deposits. That bargaining will occur with that particular Gaubura. And what they did is, I just want to show you. This is our area. This is not important. They are digging a small tunnel that is called red hole. Hardly one person can enter into that red hole. And there is no any safety measure, no anything is there. One red hole is around 1 kilometer long. The labor which are going from Gwalpara and Dhubri, they are working in that particular coal excavation place, and they are getting 1,000 rupees per day. Major accident is always happening in that particular site because of this is human right. I think I am not this is not my duty. Just I want to tell you. You can see some water is coming from that red hole that is called acid mine drainage. You can see it is very transparent, but it carries huge amount of heavy metal, like iron, zinc, lead, even cadmium, arsenic, etc. Even sometimes they're told that in Jointia Hills, some radioactive materials are also there. You can see in that abundant pit, lakhs of abundant pits are there in that particular site. That is in Nagalbibra, which has now well connected with Jointia Hills due to that excavation of coal. The coal is the third grade coal. We have three types of coal: anthracite, then lignite, and bituminous. A lignite type coals is there in case of Garo Hills. That carbon content is less. When it says that when carbon content is less, amount of heavy metal is more. In case of Lido Margarita also, lignite type coal is there. I just want to, that all the AMD, acid mine drainage, they are accumulated in that river side. You can say it is very transparent, but it is devoid of any organism. Even the insect is very tolerant to any type of pollution. They are also absent in that particular area. When you go to that particular place, within fractions of second, itching will occur in your feet. And always that villagers warn you, don't go to that particular site. We have collected that AMD from that particular site. We got one project from DVT in 2012 and collected AMD along with the fish. What we did is, after 95% dilution of the AMD, we expose. I'm visible, please.
Yes, sir.
Yes, sir. After 95% dilution of that AMD, we just expose that Sanna punctata, that Goroi mass, for 1 hour. After 1 hour, you can see the result, the toxicity of that AMD. You can see that caudal fin has gone, that is lesion-like structure appeared in the ventral surface, saddle-like lesion is found in the dorsal side. When exposure is going on, what will happen, I will show you. This is the normal scanning electron microscopy photo of gills. You know that in case of fish, our kidney is the purifier of blood. In case of fish, the main purifier of blood is gills. This is the normal lamellae of the gills. After 1 hour of exposure, what happened? You can see the gills. What is the size of the gills? Totally destroyed by this AMD. Okay, just I am going by fast. And when we got this, after exposure, what we did is, we collected the blood from that particular fish and test one. This is very easy, practical. You can apply. You can do that in your laboratory also. Just you collect after exposing the fish, you just collect the blood of the fish. I just tell you the principle. If any fish or any organism are in stress, if the stress is because of some genotoxic agent, the pollutant they can have the capacity to degrade the gene that is first appeared in the RBC, that is in nucleus, the nucleus of the RBC. If the fishes are in stress condition due to the genotoxic agent, the RBC will divide into a small RBC that is called micronucleus test. This is the first test to determine whether any genotoxic agent is available in AMD or not. The second one is, you can see this is the older slide. The second one is chromosome elaboration. The second test, that chromosome study is also a very interesting study. You can also do that. Just you have to erase the mitosis. You take the exposure fish, colchicine injection, and take the kidney or gills after, preferably kidney, after 2 hours, dissect the kidney, and you get the chromosome. we have see the chromosome. After exposure of 1 hour, the chromosome get detached, that chromatid get detached from each other. This is the third proof of that genotoxicity. That last one, that is comet assay. Actually, in that particular experiment, that fluorescent microscope is required, which is there in our lab. You have to do the microtomy, but just in that microtomy, you have to put some fluorescent dye. After using that fluorescent dye, you have to examine it in fluorescent microscope. If the nucleus is very intact, you will see this type of photograph. If gene is get disintegrated, you will see this type of shadow. If whole gene is get disintegrated, you will see that comet-like structure. Therefore, it is known as comet assay. This is the confirmatory test if any agent is having any genotoxicity. That means what I am telling is the coal deposits don't publish in the newspaper, otherwise they will grill me. The coal deposits which is found in our area, the AMD, is having moderate type of genotoxic agent. That has also published in published in Elsevier journal, that Ecotoxicology and Environmental Safety. I want to tell you one story, because all are faculty member. After completing this comet essay, just we write the paper, send it to the Ecotoxicology and Environmental Safety. Within 3 days, that paper has rejected with a comment. Just I am repeating it again. Within 3 days, they have rejected the paper with a comment that though you are working in AMD of coal mining, your experiment is right, but you fail to classify the degree of genotoxicity of your AMD. They have also advised one thing. Our principal sir can able to know, he is very, because he is from chemistry, he can able to tell that they have also indicated that the degree of genotoxicity can be done when we evaluate PAH, that is called polycyclic aromatic hydrocarbon. The amount of the class of polycyclic aromatic hydrocarbon that is found in that acid mine drainage. And immediately we rushed to IIT Guwahati. Through atomic absorption spectrophotometry, we did that how many classes of polycyclic aromatic hydrocarbon is there in AMD. Interestingly, we got 24 different classes of polycyclic aromatic hydrocarbon, out of which the most toxic is benzopyrene. I just want to tell you that compare the toxicity of benzopyrene. One person, when he exposed to benzopyrene, inhalation of benzopyrene gas for 10 minutes is, I compare. A person is smoking 10 packets of cigarette a day, or a person inhaling 10 minutes benzopyrene smoke. The problem or the toxicity or the defect is 100 times more in case of that benzopyrene than that of the smoker. This is the toxicity of the benzopyrene. When we did classify the polycyclic aromatic hydrocarbon, within 1 month, consecutively, we have published 2 papers. One in 2016, one in 2017 from the same journal. That is now the impact factor is 4.6. In addition to that, we published another 7 or 8 papers from that particular. This is the toxicity which is going on. without the knowledge of local people, we don't have any industry, we don't have any water pollution. But when that minerals is excavated from the earth crust, they are also yielding some of the very toxic pollutant. Just I am coming to the last part of my presentation. If you are not bored, just hurriedly I'll show you. That is cumulative approach. What to do? Just I have shown from my academic interest, I have shown the section 1 and section 2. What to do, how we can go for how economic approach, what is the aquaculture approach when some spelling is mistake, that A is missing. Okay. What to do, how we can able to earn something from our natural product. Just I have spelled out that freshwater ornamental fish culture and breeding, then high density fish culture using RAS bioflow, and intensive fish culture through culture that is called semi-intensive and intensive mode. I'll show you. In what we are doing, that is for the people of Assam, we are already, I told you, more than 20 species we have successfully bred in our center. Just I want to give you some glimpses. That pipe fish, that is called microfish Dakota, that is the only pipe fish which is freshwater pipe fish found in Northeast India, which is congenial to seahorse, and now it is the near threatened species which has been
Sir, you have been, your mic has been muted.
Sir, your microphone is off.
Sharma sir.
Sharma sir, your microphone is off.
বাপ্পা আনমিউট করতে পারবা? বাপ্পা আনমিউট করা যায় না?
মাইক্রোফোনটা হ্যাঁ।
না স্যার শর্মা স্যার তো আউট হয়ে গেল।
হ্যাঁ না আমি বলেছি আমি।
now it is okay.
Yes, sir. Yes, sir.
Just I have to share again this. Can I get? I am sharing it again. Is it visible now?
Yes, sir.
Yeah.
Yes.
This is that our student has done some technique. What they did, they can able to take the photo from the take the egg from the brood pouch of the male, and after taking photograph, they can able to insert it again without damaging the egg. In that way, you can see this is all the larva, different stages of the development. Just I want to show you one video how they are. Is it visible?
Yes, sir. Yes, sir.
You can see this the species which swell the abdomen, this is female. During that process, that female can able to inject the unfertilized egg into the brood pouch of the male. Just you show the video for 30 seconds. Is not possible in the surface of the earth. Explain with suitable example. Okay, is it interesting?
Yes, sir.
Oh yeah. Okay. Just I want to. We have published one paper in the month of September 2019 on that particular species. Why I am showing that paper? Because how important our diversity is. We have published it in Journal of Applied Ecology, that is from Netherlands. Immediately after, when it appeared in the journal, I got 2 mail from National Geographic Channel, because these people are very much enthusiastic to have a short film on that particular species, and they're supposed to they send me the mail, supposed to come in the month of March 2020, but COVID-19 has shuttered everything. Still, we are waiting for the situation to be normalized. Hopefully, they will again come to have one small film on that particular species. I am audible now?
Yes.
Already I told you that Sanna Stewarty we successfully bred in our center. I will show you that is male and female can be differentiated like that way. You can see this is the larva. Natural breeding. You just put some Eichhornia, that is called Meteca, and feed the earthworm. Only you keep it in a small cistern. No oxygen is required. Put some Eichhornia. Give only earthworm. During the breeding season, that is April, May, June, they will breed automatically. And the male fish, they are guarding the brood, all the fingerlings. If you disturb that species, they will engulf, they will eaten up all the larvae. This is the main problem. So you have to give. That can be possible. That can be done in home also. This is the development. And Sanna and Rao, this is also very lucrative species for ornamental fishes that can also be bred in our aquarium. You can see the larva. This is lots of slides are there. I'm skipping that slide. And we have also successfully bred the Divariocupinetas. This is also very lucrative as well as ornamental as well as food fish. I'm showing this one, Denudendula. These are the embryology. Our student has taken this photograph. Only that why I am showing that our undergraduate student, they can able to do these things. They are taking this photograph with simple microscope with their mobile camera only. These are our works which I want to. This is another species that is endemic to Zoanthia hills, Pythia salineus. Now it is vulnerable. We successfully bred that particular species. Now our target is to make it as a mass cultivation of that particular species so that we can able to give it to the farmers. This is the target. In addition to that, we are also doing Pabda and Magur. And this year, though it is pandemic year, but we successfully bred more than 2 lakhs of Pabda in our center in addition to Magur, because that season was very good for Pabda breeding. And another species that is called Chinox nobilis, that particular species is also enjoying a high value in international market, is around 10 to 15 pound per pair. This is also live bearer, that male keeping the egg in the mouth. I'll show you the video how they are doing it. Just 1 minute video. This is the courtship behavior. After courtship, that is it visible?
Super.
Yes, yes.
After courtship, that female will release the egg from the mouth and male will engulf it. But it will take 1 minute, please. Because I don't have that capacity, we cannot edit that photograph. Just bear with us. You see how they are searching a particular environment so that they can able to release the egg from the mouth. Immediately after releasing from the mouth of the female, the male will take it up, and male can able to keep that particular egg for 25 days without eating, without opening the mouth. After releasing the larvae, the male dies. Just a minute. I want to show you how they are searching the good opportunity so that they can able to release the egg. You just see carefully. Now they will release. Please see carefully. They are in the mood to release the egg.
বন্ধ করছি। হ্যাঁ, এখন
Just a minute. Please, you see now that female is about to release the egg. You see. Okay. In that way, this is the natural breeding. And just I want to tell you one thing. Fishes will grow in your pond when that plankton production is maximized. That when plankton is more, then it will grow more. Just I want to skip this thing, that is not necessary. I just want to tell you that is the Rooftop Bioflow. Actually, it costs around 20,000 rupees. You can able to erect it in your rooftop, and 5,000 liter capacity where you can able to rear 1,000 anabas, that is cow mass. and due to that waste which is produced from that particular bioblock, you can use to cultivate some that vegetables. We are in our center, we are going to have one RAS that is called Recirculatory Aquaculture System in our center. That is, in a small pond, 1 hectare pond, we can rear 7,000 fingerlings. But in that particular RAS system, with a 90,000 liter capacity, we can able to rear 7,000 fingerlings for 3 months. This is a very new technique, which is very lucrative also in addition to that vegetable cultivation. This is all about that what we are able to do, that in addition to that pond culture, we have bioflow, RAS, as well as ornamental fish culture and breed. The last one, I just want to tell you that there is the alien species. Don't actually most of the our bills, they are having this type of species that is called crocodile fish. This is not our species. It has already infested most of our natural resources. Just I request you, if you see this type of species that is called Hypostomus placostomus, South American one species, that is usually it was bringing it as a ornamental species, but somehow it escaped from the aquarium, entered into our natural resources, which is creating havoc throughout the Assam. So just kill it and tell all the farmers to kill it. It is very dangerous for our ecosystem. Thank you very much.
Thank you, sir.
Yeah.
Thank you, sir.
Yeah. Thank you.
Now the session is open for question and answer. Hello? Anybody? If anyone has any question or any point, please put it in the chat box.
প্ৰফেছৰ শৰ্মা, মোৰ এটা বেলেগ প্ৰশ্ন এটা আছিলে।
কওক।
তাৰমানে এই যে আপুনি দেখালে না ৰুফটপটো, এনেকুৱা ধৰণৰ ৰুফটপ এইবিলাক ক'ত পায়?
আপোনাক মই ফোন নাম্বাৰ দি দিম, আপুনি লৈ ল'ব, তাতে গৈ বনাই দিয়ে একদম।
নেকি? আচ্ছা,
মই আপোনাক অলপ পিছতে শ্বেয়াৰ কৰি দি আছো।
আচ্ছা আচ্ছা, আৰু এটা কথা আছিল, এই যে আপোনাৰ লগত এবাৰ পাতিছিলো মই, এই যে যিটো এইমাত্ৰ দেখালে কি RF চিষ্টেম নেকি ক'লে?
ৰিচাৰ্কুলেটৰী একুৱাকালচাৰ চিষ্টেম। আমি একচুৱেলি এইটো আৰম্ভ কৰিছো আমাৰ চেণ্টাৰত। কিন্তু মানে কওক।
এনেকুৱা এটা সৰু ছেটআপ ফৰ এডুকেশ্যনেল পাৰপাছ আমাৰ ষ্টুডেণ্টৰ কাৰণে বা আমাৰ এই ওচৰৰ মানুহৰ কাৰণে আমি ছেটআপ কৰিব পাৰো নেকি আমাৰ কলেজত? হাউ কষ্টলি ইট ইজ? বহুত কষ্টলি নেকি?
আমি যিটো বনাই আছো এইটো কষ্টলি হয়, এইটো প্ৰায় 10 লাখ। আমাৰতো বহুত ডাঙৰ, তাৰমানে আমাৰ মানে আধা সৰু কৰিব পাৰি।
আচ্ছা, তেনেহ'লে এইটো পিছত আপোনালোক আৰু ভালকে কথা পাতিম। ঠিক আছে।
Hello?
Yes.
Hello, sir?
Yes. Yeah.
Now, Sushmita is not there. Now, vote of thanks by Dr. Samuel Sheikh. Samuel?
Yes, madam. Yes. Thank you, madam. I, on behalf of the Department of Zoology, BN College, Dhubri, convey the vote of thanks to Dr. Dhruva Chakraborty, Principal Sir of our college, for allowing us to organize this webinar. Thank you, sir, for your active support. Next, I convey my sincere gratitude to Dr. Dandadhar Sharma, sir, the resource person of this webinar, for his lively and excellent deliberation on the topic, particularly the videos you have shown, sir. It is very interesting. Despite his busy schedule, he has attended the program and made this program a successful one. I also convey my sincere thanks to the IQAC coordinator, my respected colleagues, the technical person, GTech, and others for their entire support in organizing this event. My sincere gratitude goes to all the teachers and the research scholars of different renowned institutions who have showed interest on this topic and joined the event to make it successful and dignified one. Last but not the least, I convey my heartfelt thanks to all my dear students who have joined the program to make it a successful event. Thank you all once again. Thank you.
Sir,
Over to you, madam.
now the regarding feedback link, it will be available now today and active for 24 hours. It will also available in our college websites and your registered email ID also.
Yeah, may I add just a last sentence?
Sir. Okay.
So thank you very much, Professor Sharma. So it is a fascinating area where you are actually doing your research, and the way you have you are doing your research, you and your team actually, it is very fascinating, and I found it very fascinating, and I am sure that it is helping the farmer community and the entrepreneurs of Assam as well, and hopefully in the near future some of our students, some of the participants from here may become entrepreneurs in near future. So with that note, I would like to again thank you and hand over it to Mitali.
Thank you very much.
Now, sir. Thank you, sir. Thank you.
Okay.
Thank you once again.
Okay.
Now I complete this session.
Okay.
Thank you.
Thank you.
Didi, congratulations. Hello, sir. Professor. Hello. Professor. Hello. Professor. Hello.
Ồ, bây giờ sẽ được